Hi everyone. So for this video, I thought we would just go over the study guide. We didn't get a lot of time to work on the study guide in class and get feedback from the teacher or from myself. So I thought I would go over the study guide with you instead of just like giving you an answer key. So the first section here. So for this video, we're going to go over let's do the first 2 pages. Okay. Okay, first we'll talk about meiosis. And what is our goal of meiosis? So our goal of meiosis, we are trying to make gametes. And so then your question might be, well, what is a gamete? Okay, gametes are sex cells, okay, or they are cells specialized in uh, passing on genetic information to offspring. Okay. Examples of gametes would be sperm or egg cells or pollen. Okay. And something interesting about gametes is that they are haploid. This means that they have one of each chromosome, or you can say one allele for each gene. Quickly, I will show you haploid. Sorry about that. My toolbar is not on my screen. Okay, so this would be a haploid karyotype where we have one of each chromosome and one sex chromosome. Okay, and this is different than a diploid cell, which has 2 copies of each chromosome, one from the mother, one from their father. Okay, so if we go back here, now that we learned about what gametes are, let's talk about somatic cells a bit. So somatic cells, these are our body cells, okay? Cells that are not sex cells. Okay. Examples would be our muscle cells. They would be our skin cells. Okay, any cells that are not specialized in passing on genetic information. And these cells are diploid, okay, meaning they have 2 of each chromosome, one from each parent. All right. So now let's talk about, so now let's get on here. Why is it important that gametes are haploid? Why is it important that gametes only have one of each chromosome? Well, we did talk about this before. It's important for them to have one of each chromosome. That way, when the cells are fertilized, the resulting cell is diploid. Okay? So as you can see here, we have a haploid egg cell that's being fertilized by a haploid sperm cell. So one copy of each chromosome. When these come together in fertilization, it produces a diploid zygote. So now we have 2 of each chromosome, one paternal and one maternal. So if we go back here to our study guide and say, why is it important that gametes are haploid? That way, when the gametes are joined in fertilization, the resulting zygote, or you can say offspring, is diploid. Okay. So what is fertilization? This is when our gametes are joined. Okay. You can say the joining of gametes, or you can say when the genetic information of gametes is combined. Okay, that would be our fertilization. Okay, so we talked about meiosis. Our goal is to make gametes. which are our reproductive cells, and we talked about what gametes are. And now let's talk about what is our goal of meiosis 1. Okay, so if we go to our diagram of meiosis, we have meiosis 1 and we have meiosis 2. In one of these, our goal is to separate homologous chromosomes. And in one of these, our goal is to separate sister chromatids. So go ahead, look at the model, and try to decide, in meiosis 1, are we splitting homologous chromosomes or are we splitting sister chromatids? So you should have realized that we are pairing up here the homologous chromosomes, long with long, short with short, and then we are going to separate them into separate cells. So in meiosis 1, we are trying to separate the homologous chromosomes into 2 separate cells. So meiosis 1, to separate homologous chromosomes into 2 separate cells. And our goal of meiosis 2 is now we have a haploid cell. We have 1 long chromosome, 1 short. So we're not separating homologous chromosomes because we only have 1 of each, right? In meiosis 2, we're going to be separating the sister chromatids, right? So this chromosome here has 2 sister chromatids attached to the centromere. We're going to be splitting those sister chromatids into separate cells. Okay. However, sometimes we have mistakes in this process of separating our homologous chromosomes or separating our sister chromatids. This is called non-disjunction. I'm on slide 54. So if we could have non-disjunction where in meiosis 1, our homologous chromosomes do not separate properly. What is the result? You know, half of our gametes, if it happens in meiosis 1, half of our gametes will have an extra chromosome, and half of our gametes will be missing a chromosome. The same thing can happen in meiosis 2 with the sister chromatids. If our sister chromatids fail to separate properly, then we'll end up with one of our gametes having an extra chromosome and one of our gametes missing a chromosome. Okay. So let's talk about this. Sorry. Non-disjunction, what is it? It's when the homologous chromosomes or the sister chromatids fail to separate in meiosis. Okay. For example, we can just put, so what is the result of this? Okay. What is the result if we have gametes with an extra chromosome or missing a chromosome? Our result would be either a trisomy or a monosomy. So, one of the misconceptions that I noticed in the Calyx gizmo, the Calyx the cat gizmo, is people are like, oh, Calyx the cat would have some cells missing a chromosome and some cells with an extra chromosome. No, that's not true. Meiosis is happening in his parents, in Calyx's parents, right? Calyx will inherit just one of these gametes. So he will either inherit a gamete with an extra chromosome or missing a chromosome. In Calyx's case, because he had an extra chromosome, he inherited a gamete with an extra chromosome. Specifically, he inherited an extra X chromosome. So he had a similar mutation like this. So he had a trisomy where he got an X from the mother and he got an X and a Y from the father. So the father's gamete had an extra chromosome. So any of these mutations, these trisomies and monosomies, result from the fertilization of a gamete that was the result of non-disjunction. So the fertilization of a gamete which has an extra chromosome, extra or missing chromosome, results in a trisomy or monosomy mutation. Okay. And this, we don't really have to answer that. Okay, great. So we're done with the first page. Let me know if you have any questions. You can always leave comments if you have questions. So now let's talk about homologous chromosomes. What is true? What is false about them? Homologous chromosomes, are they similar in length? Yeah, they are. Do they code for the same genes? Yes. So if we look at these homologous chromosomes, if this one does hair color, yes, so does this one. Okay? They are homologous. They have the same genes. So like Calyx the cat, this had fur color. So the other X also has fur color. Okay? So they do code for the same genes. How are those genes on the same positions or location on the chromosome? Yes, they are. And that's important for crossing over. When we look at crossing over of homologous chromosomes, my blue gene here is in the same spot on my homologous chromosome. This green gene is on the same spot on my homologous chromosome. But do they have the same alleles for each gene? Not necessarily. They can, but not if they're homozygous, but not always. So this one has a capital R, and this is a lowercase r allele. They have different alleles. Okay. Okay. Now, features of meiosis that contribute to genetic variation. This was also on the Calyx the cat gizmos, one of your free response. One feature is crossing over, and the other one is independent assortment. So crossing over is when genetic information is exchanged between homologous chromosomes or between the maternal and paternal chromosomes. I would pause it, but I don't have my toolbar today to pause it while I type. So it's when genetic information is exchanged between homologous chromosomes, the maternal and paternal. When does this happen? We talked about it happening in metaphase 1. However, if you did put prophase 1, that is okay. Independent assortment. Independent assortment is saying, okay, our homologous chromosomes or our sister chromatids can be separated in either direction. So for example, if I look at this cell here, both of my paternal chromosomes could go in the same direction to the left, and both of my maternal could go to the right. Or I could have a paternal this paternal could go to the left, and this paternal chromosome could go to the right. So it's kind of like random. It's independent. They're independently assorted to one another. This one is independently assorted from this one. So independent assortment, that chromosomes can separate in either direction during anaphase. Okay, so when does this happen? It happens in anaphase 1 and anaphase 2. So in anaphase 1, it's the homologous chromosomes that are doing this, that are separating. And in anaphase 2, it's our sister chromatids that can go in either direction. Okay, now, and so these are the two ways that we can get genetic diversity through meiosis, but why is it important? We talked about because if there is a catastrophe, for example, a new disease, right? If we have genetic variation within the population, there's a higher likelihood that there will be individuals that have the genes to survive that catastrophe. So if there's a catastrophe, for example, a new disease, If there is high genetic diversity, there is a higher probability that a segment of the population will survive. If there's a catastrophe, for example, a new disease, if there is high genetic diversity, there is a higher probability that a segment of the population will survive. Okay, so that is the first 2 pages. I said that that would be our video, our first video, and I hope it was helpful. Bye.